As Eamon said, I'm going to try and discuss diet and exercise and our attempts at teasing apart the effects of them individually, and if we are ever going to be able to look at diet and exercise individually. So why are we interested in exercise? Well, as Paul mentioned earlier, almost every aspect of modern lifestyle will have an effect on your gut microbiome, and nothing more so than diet and exercise. So exercise affects your gut transit time, so then how long your food sits in your diet, in your gut, so therefore affecting the microbiome. It affects your appetite, it can affect your mood, and in fact a lot of athletes, once they stop training, suffer a lot of mental health problems. It also affects your anti-inflammatory tone, your immune system. And whether or not we'll ever be able to tease apart exercise and diet, a lot of us will know when people start an exercise program, they either go on a strict diet in conjunction with it, or subconsciously they start rewarding themselves. So you go on, I've gone to the gym for 2 hours, now I deserve a McDonald's, or I've gone to the gym for 2 hours, so now I can't eat for the next 6 hours because I'll undo all my good work. And another thing that happens to elite athletes when they stop training as well is that a lot of them get eating disorders because they want to counteract the loss of their extreme exercise by controlling their diet in a lot of ways. So whether or not we'll ever be able to truly tease it apart is something that we haven't yet figured out. So no better way to look at something like this than to have a case study. And there's very few studies on the effects of exercise in the microbiome in humans. And I guess in APC and Teagasc we're very fortunate that we had the first such study in 2014. And we've recently followed up with another study in gut also in 2017. So we had the Irish international rugby team at a pre-World Cup training camp in 2011. And for those of you unfamiliar with the rugby team, there are two very distinct type of players in a rugby team. The forwards, quite strong, but they do have a high BMI. The backs are made for speed, so they're very lean, they're very fast, low BMI. So for this reason, and to eliminate BMI as a confounder in the study, we had two control groups, a low BMI control group and a high BMI control group. And basically we threw the kitchen sink at this study. We had urine samples, fecal samples, and you'd be surprised at how difficult it is to get fecal samples out of anybody, particularly athletes, so very reluctant to give fecal samples. They'll give you blood far quicker than feces. We took diet in food frequency questionnaires, and we had DEXA scans, and we did full metabolomics, shotgun metagenomics, compositional metagenomics, all clinical blood testing, and as I said, DEXA scans. So the physical characteristics, as you'd expect, despite the high BMI of the athletes, they had a higher percentage of lean body mass, and they were just basically physically better and fitter. Surprisingly, they had a lower inflammatory tone compared to the controls, despite the tough regimes that they put their bodies through. When we looked at their diets, the athletes, this is a correspondence plot, so the athletes here are in black, so they separated quite distinctly from the controls, which are the blue-red lights. And what was driving this separation was things like sports drinks, which you probably would expect from athletes, but also healthy, good, energy-efficient foods, whereas the controls were branching with processed foods, sugary foods, things that we really should be cutting down in our diet. And we want to draw attention to this one athlete here, who's going to pop up again in a minute. This particular athlete clustered with the high BMI groups always. When we went back and looked at this athlete's food frequency questionnaire, they ate no fruit, no vegetables whatsoever. So it kind of alludes to the opinion that you can't out-train a bad diet, even if you are an elite athlete. And this exact same athlete is going to come up again in a minute, and we'll talk about him again. Paul, if you know that we work in offices side by side, the exact same slide, so I could just skip over this diversity analysis, but this became very important to us as this particular study proceeded. Another thing that we had to do for the athletes was to find out how could we compare the fitness levels of an elite athlete with a non-exercising control group. And we couldn't, the standard way you'd compare exercise between groups is ask them how many times a day do you go up and down the stairs, how much gardening do you do, how many steps do you take in a day. We couldn't necessarily ask athletes that, nor could we ask a control group how many lengths of a pitch did you run, what kind of weights did you push today. So we came across this paper of endurance athletes that they had measured their fitness levels using creatine kinase. Creatine kinase is an enzyme that's released from muscles post-damage. It's used in a clinical setting to assess the levels of MS or for cardiovascular illness. But in our athletes, it's released from muscles post-intense exercise. And you can see here that the athletes had much higher levels of creatine kinase compared to either control groups. And we were very happy to use this as a proxy when we looked at the outliers in the low BMI control groups. This particular guy had just run a marathon in the week previous and had the high levels of creatine kinase in his blood. And these other two outliers were amateur GAA players, which is a sport here in Ireland that you'd practically train to professional levels. So we kind of thought that this was quite a good proxy for fitness levels in our groups. And it's not surprising that the creatine kinase levels are so high in rugby players. For those of you that aren't familiar with rugby, this is the kind of action that goes on in a rugby match. So you can see there is a lot of muscle damage that's undergone. So going back to the diversity. So as Paul said, that some individuals we found that have very high diversity levels. And whether or not this level, such high levels of microbial diversity are a good thing or not is yet to be discovered. But for now, all the literature is pointing to the fact that high diversity is good. And in every single one of the 5 metrics that we used to measure microbial diversity, the athletes were significantly higher than all of the control groups. When we looked at beta diversity, we saw no separation in the control groups. So there was no separation of beta diversity based on BMI. And when you put in the athletes, they were distinct from the controls. And here is this guy, again, going up with the high BMI control groups, the guy that ate no fruit and veg. So we see that, again, you can't out-train a bad diet, is what we're kind of taking from that. What we wanted to figure out was what was driving this microbial diversity. When we went back to our clinical data, we had 180 pieces of clinical data, whether it be from the bloods or the food information that we had. And we correlated the microbial diversity against every single one of these pieces of data. And 2 things popped up. Microbial diversity correlated with protein. And when we went back to the diets again, the athletes were taking a larger percentage of their energy from their diet was coming from protein. And when we delved even further into this, it was in the form of whey protein. And the other thing that was correlating with this diversity was CK or fitness levels. So basically microbial diversity in the athletes was correlating with either or protein and fitness levels. So we went on further. So this was published in Gut in 2014. And using the same data, we went on to further characterize with using shotgun metagenomic analysis and full unbiased metabolomic analysis. And the first thing we did was compared our shotgun data to our compositional data from a taxonomic point of view. And here, this is what you get when you look at the shotgun profile of all, and this is the compositional. Overall, the taxonomic profile of both were quite similar. The blue radials here, you see there's a lot more in the compositional side. These are poor identified or unidentified species and genus. So you see that the shotgun is much better at getting a high percentage of identification. What was the most interesting thing that popped up from us was that Akkermansia was enriched in both the athletes and the lean control groups. And this has been published many times, particularly by William DeVos's group in recent years. The Akkermansia has been associated with a lean phenotype. So the fact that it's enriched in athletes and our lean controls was a very good thing. From a functional perspective, you see the similar to the compositional and the alpha diversity that we already saw. The athletes were significantly more diverse from a functional perspective than the control groups. And again, when we separated them out on a PCOA plot from a beta diversity point of view, the athletes separated out from both the control groups. So they were functionally distinct and more diverse than the controls. What was driving this functional diversity again? So the athletes groups had highest abundances in things that are beneficial to the host and the ability to harvest energy from their diet. So carbohydrate biosynthesis, cofactor biosynthesis, energy metabolism, and also creatine kinase, bilirubin, and energy intake. So things that are going to help the host work. Whereas the control groups, whether it be driven by a deficit in their nutrition, had things that were linked to getting sugar, protein, and carbohydrates. So we kind of postulated that the control groups weren't getting enough of these nutrients from their diet and the microbiome had to work hard to produce these for the host. From a metabolomic perspective, this was done in collaboration with Imperial College London, Professor Elaine Holmes, and they actually commented that they have never seen such a distinct separation in a non-disease cohort. So the athletes and the controls, again, completely separate in untargeted metagenomics, both from urine, which would be reflective of the host metabolism, and the fecal water, which is reflective of the microbial metabolism. So both very distinct. And again, when we delved into this in targeted metabolomics, what we saw is that the short-chain fatty acid profile was unique to each of our cohorts. So basically the short-chain fatty acid profile was enriched in the athletes. Short-chain fatty acids have a lot of beneficial properties to the host, including they're anti-inflammatory, they improve energy harvest from your food, they can pass the blood-brain barrier and improve appetite. So the fact, again, that these were increased in the athletes was only a good thing. When we correlated these short-chain fatty acid profiles back to all our metadata, what we saw was two distinct blocks appearing. So acetate, propionic, and butyric acid, three of these short-chain fatty acids, were correlated with things that come from your diet, so protein and fibre, whereas isobutyric, isovaleric, and valeric acid were being driven by bacterial diversity. So perhaps these three short-chain fatty acids can be driven by diet, whereas these three are more involved with your microbiome. So the highlights from a metabolomic point of view, in the athletes, they were increased in products of protein metabolism, namely TMAO, carnitines, trimethylamine, and 3-hydroxyisovaleric acid. These are all very new to us because none of us are kind of high up in metabolomics, and a lot of these have never been published before as being increased in athletes, so it's kind of a jumping off point for us, and now we're going back to kind of try and individually tease out what the true meaning of each one of these effects is. Furthermore, athletes had an increase in the metabolites of muscle turnover, as you'd expect, so the creatine being released in the muscles and 3-methylhistidine. Athletes also had increases in the metabolites involved in vitamins in recovery, so glutamine, lysine, and 4-pyridoxic acid. So basically, the athletes were primed for better use of energy from their diet, which is kind of what you'd expect from an athlete. The conclusions: distinct functional and metabolomic profiles. Pathways were related to fundamental metabolic function were higher in the high BMIs. On the contrary, athletes, the metabolites were primed for tissue repair and to harness energy from the diet. And notably, the low BMI group was always most closely related to athletes, which kind of would lead you to the conclusion that small changes in your diet and your exercise can perhaps improve your metabolic and functional profiles. So where did we go from here? So we knew that two things were driving diversity and gut health in the athletes: protein intake and or fitness. So could we somehow tease these apart in a group of non-athletes? So from there we went on to design a human intervention trial with 3 groups. So we had an exercise-only group, a group that we had exercise and added 24 milligrams of whey protein supplement to their diet, and a whey protein only group that didn't exercise and just had the introduction of whey protein on a daily basis. It must be noted that all of these individuals were required to be what we deemed couch potatoes, so they were not undergoing a fitness program and were fairly unfit. So we had great compliance, 99% of compliance with the whey protein intake, despite the fact that it was banana flavoured whey protein that we were using. And the group that had the whey protein only were offered a fitness program after the initial 8 weeks to so that they wouldn't feel like they were losing out on a fitness program. So very briefly, in the nature of time, this work is currently finished and under review at the moment, so it's still all new. So the clinical findings at the baseline, the 3 groups are homogeneous, so there was no differences. So that was good from an analysis perspective, in that we were working off the same baseline. When we looked at the 2 exercising groups, their gym activity was not significantly different. So again, we're looking at homogeneous groups, and it wasn't different levels of exercise that was affecting our results. Post-intervention, as you'd expect, the exercising groups got fitter, lost more body fat, and gained more muscle. Whether it be surprising or not, the rest of the results were quite limited. So we recorded their diets with food frequency questionnaires. We were instructed them not to change their diet. It's very hard in a human study to control diets or to tell people exactly what they should eat. So the conclusion we came to was that they should just continue on with their normal diet and just introduce the whey protein. When we looked at the changes over the start and the end, pre and post intervention, there was no significant differences in pre and post intervention in any of the groups. So for us, that was a good thing. It wasn't going to be a change in diet that was driving any changes that we saw. When we looked at the change of diversity pre and post intervention, for this we only did shotgun analysis, so we were able to look at archaea, bacteria, viral and functional pathways. There was no differences in any of the groups pre and post intervention in any of those particular categories. Nor was there any differences at the functional level or at the metabolomic level. When we looked at the beta diversity, the only differences that we saw, so this is, bacteria, viral, archaea. The only differences that we saw was in the viral species when we introduced protein. So the top group along here is just exercise plus protein, and just protein. And in the 2 groups here in the middle that had protein introduced to their diet, there was a distinct separation of the viral species. We have yet to figure out the cause of this. Somehow we think that the introduction of the whey protein, whether it be through phage that is in the whey or something like that, is driving this distinction between the clusters. But it's an interesting observation that we're going to have that definitely warrants further investigation. So in conclusions, exercise groups had increased fitness and reduced body fat, but this did not induce a significant alteration in gut bacteria, viral or functional or archaeal diversity. And it also did not drive any changes in the metabolome or in the functional section of the bacteria. So functional activity of the gut microbiota does not appear to be significantly manipulated by short-term intensity exercise or whey protein supplementation. So the acknowledgments, this work was all done by 3 excellent students, Owen, Wiley and Siobhan, and is driven by 3 outstanding PIs. And we also want to always thank all our athletes, both elite and not, for kindly donating samples.
Thanks very much.